Welcome to this introduction on a recent paper published in Nature Communications entitled A Systematic Comparison of Tools Used for m6a Mapping from Nanopore direct-RNA Sequencing. And this is really important because Nanopore direct-RNA sequencing data is the first that allows for transcriptome-wide m6a mapping, as opposed to the limitations on technologies that relied on short-read next-generation sequencing. But before we dive into it, let's ask a basic question. What is m6a, or what are these m6a modifications? Well, it's a specific chemical modification on the base pair adenine, as shown in the diagram here. It's mediated by the proteome, specifically by writers such as metal 3 and erasers such as FTO. m6a modifications are important because they interact with the proteome through something called a reader protein. An example of a reader protein is the HNRNP protein. These readers can then mediate RNA metabolic processes such as mRNA export and decay. M6a is a type of RNA modification, one of 150 that are thought to exist within human RNAs and in other mammalian species. M6a, though, is the most abundant of these and well studied, which is why it's one of the first that has these whole transcriptome-wide options, or this whole transcriptome-wide detection option available. Previously, before the advent of nanopore sequencing, m6a sites would be detected via short-read RNA sequencing methods. And there would be two particular strategies that were generally implemented in order to detect these m6a sites. The first is shown here on the screen, which is a workflow based on immunoprecipitation, where an antibody would be utilized in order to detect m6a sites. The second strategy, which is not shown on the screen, would rely on endonucleases or protein-based digestion methods that would specifically identify these m6a sites, digest or modify in a certain way that could be followed up by short-read sequencing and detection. And then based on the specific strategy, you would undergo a computational pipeline, as shown here in panels B and C, that allows you to, based on the union of the different fragments, or based on whatever the endonuclease digestion suggests, allows you to identify a particular peak associated with the base pair that you assume to be m6a modified. These strategies rely on high coverage in a particular region, and therefore it takes a large amount of sequencing data in order to accurately identify a given site. Nevertheless, for the purpose of this systematic comparison between tools, these kinds of strategies, including ME-RIP-seq as shown here, but also m6a ref-seq, MI-CLIP, and MI-CLIP2 were utilized in order to generate ground truth data sets for comparison and further analysis. Let's step back and get a little bit of context on this nanopore technology and why this transcriptome-wide m6a mapping is available now. Nanopore in 2018 released the first paper demonstrating direct RNA sequencing, which means that their system is examining the RNA specifically, which therefore allows it to detect these RNA modifications. It's based on an electrical current that changes and fluctuates as the RNA moves through the pore. These electrical currents can be utilized in two different ways in order to detect these m6a modifications. The first strategy essentially breaks up the electrical current signal itself into different events, as shown here, corresponding to different base pairs. And these events are then directly analyzed. These signals are directly analyzed based on whatever data can either be pulled out from them or their general shape and structure, or as you might call it, squiggle analysis. You examine these and try and figure out whether there's differences between what you expect, what you canonically expect an A to look like, and what you're seeing. And if that difference is great enough, you can classify it as m6a modified. The second strategy is a little bit more downstream. After the squiggles and the electrical signals have been converted into base pairs, you can examine this, compare it to an unmodified set. We'll define an unmodified set in a second. And if there are errors in a particular position, you can assume, or you make the assumption, that those errors resulted because of an m6a modification that made it more difficult to read at that position. So these errors could be either mismatches or insertions or deletions or other typical mutations that you might see in an alignment, or it could be simply a variability in the actual base pairing quality at that particular point. So that could be based on the phi-red score decreasing by 5. The phi-red score itself is a metric for the confidence that the sequencer has in a particular base call that it's making. So in that case, you don't even need any error or like an adenine switching to some other base pair. You just need an adenine with a confidence of 20 versus adenine with a confidence of 15, which is a lower quality. The unmodified base set here is basically a data set that does not contain these m6a modifications. And in this particular study, this is the sort of standard, this is done by knocking out the reader proteins, specifically metal 3. And so in this study, they generated one particular data set with mouse embryonic stem cells with metal 3 knockout and wild type, and utilized the metal 3 knockout in this capacity where it's assumed that because the writer is knocked out, there will be no m6a modifications. And this was actually verified using known mass spec techniques and otherwise in the paper. They also performed this analysis on 2 publicly available data sets and these comparisons between tools. These data sets, one of them was a similar organization, also in mouse embryonic stem cells, wild type metal 3 knockout, and another was in an entirely different species, the plant species Arabidopsis, and mutants defected of an analogous m6a writer, VIR1. Other notable panels in this figure include C, which describes that the knockout actually had a higher base calling quality. There was an easier time moving through the actual pores and reading the data. The knockout had a slightly higher quality than the wild type. Not a significantly higher, but slightly higher, which the authors indicated may be the result of the knockout without the m6a modifications being easier to read, or having basically less confounding squiggles, which corresponds to the sort of hypothesis that the m6a modifications confound with the electrical current and make it more difficult to perform alignment features, per this hypothesis that m6a can be detected through errors within the actual downstream processing. Alrighty, so they looked at 10 different tools, but the inputs, the outputs, and as we mentioned, the overall strategies differ. whether they used statistical tests or machine learning to analyze the squiggles or clustering, or how they analyzed the alignment features, that was different between the 10 tools as specified here. And therefore, there were two different kinds of comparisons. One with single mode tools, which were defined to have no control sample, so they did not have this unmodified, and compare mode tools, which used the control samples. Certain tools only work in compare mode, certain tools only work in single mode, some work in both. And this here is basically the figure 2, the summary panel of their results for compared to the ground truth, which I mentioned is a combination of these different traditional next-generation sequencing approaches. And some of the high-level results, without diving into each panel specifically, are that m6a net specifically outperformed all of the tools for m6a detection capacity, as can be seen in the pink line here. The ranking between these different tools for precision remained consistent for the different validation sets. So in essence, whether you utilize the me-RIP-seq as your ground truth, the m6a ref-seq, the mi-clip, or the mi-clip2, the ranking remained the same across the different tools, and m6a net stayed on top. The tools that had higher precision, such as m6a net, also had a higher enrichment around the stop codon for m6a modifications, which basically corresponds with biological presuppositions and sort of the role of m6a modifications in RNA metabolism. It also noted the sort of high frequency that the tools with the higher precision sort of identified the GGACU consensus sequence, or that specific consensus motif, which also corresponds to biological presuppositions about where m6a modifications are most common. It was also revealed that there were specific motifs, namely GGACT, GGACA, GGACC, and AGACT, which all of the different Oxford Nanopore, all of these different callers performed better on. They, with much greater frequency, detected m6a modifications within these motifs precisely. This whole entire analysis, which was performed specifically on the datasets generated by this group, was repeated with the published datasets. However, as I mentioned, the structure of those two studies, the organization was very, very similar. What was revealed in this analysis when redoing this with the published dataset was that that particular metal-3 knockout sample actually wasn't a complete knockout according to the mass spec validation. They didn't perform mass spec validation, but according to their calls with the single mode caller on the control sample, which, since it had metal-3 knocked out, theoretically shouldn't have any m6a modifications. But they found that approximately 40% of the m6a modifications were present. This led to the tools that relied on the control sample with the comparison to perform worse, suggesting that you really need your control sample to have no m6a modifications in order to make that comparison meaningful. They also performed this analysis with a second published data set utilizing the plant samples, and ultimately all of the tools performed worse, but especially tools that relied on these machine learning algorithms, so especially m6a net and MINES. And that's because these tools were trained on human samples, which have different dominant motifs for where m6a modifications are present when compared to plant samples. And so in order to utilize these tools, they need to be trained on new ground truth data sets, in essence, in order to make them more applicable to these more distantly related species. The study also goes through a wide variety of other questions, both in the remainder of the results section and in the discussion. These include, how do you set the parameters to balance recall and precision when compared to ground truth? What is the effect of the metal-3 knockout on recall and precision, specifically for the single mode tools? So do the single mode tools call m6a modifications when you give them the metal-3 knockout? And is there a particular bias for the kinds of motifs and positions that they call for that? How does m6a detection capacity change based on motif? As I mentioned, there were these 4 dominant motifs, but there's more discussion and analysis on exactly what is mediating those changes. How do tools perform on absolute m6a methylation quantification? So ultimately, what all of these tools are calculating is a particular quantity that is a percentage suggestion for how likely it is that a particular site is m6a methylated, as opposed to an absolute quantification of m6a methylation at a site. So there are only 3 out of the 10 tools which attempt to perform absolute quantification. There's discussion on the particular performance of those tools to the task when compared to the ground truth. How is m6a detection impacted by sequencing coverage at a particular genomic region? What is the agreement between all of these different tools? And what are the limitations with the definition of ground truth as presented in this study? All of these are important questions, and I'll make sure to link the article below if you'd like to explore in more depth. Thank you very much for listening.